Connor, did you read or hear about the astronauts who spent 9 unplanned months in space?
Yes, I did. Butch Wilmore and Sonny Williams, and they're just back on Earth
So
in
exciting.
March. It was brilliant, wasn't it?
Yeah.
There you are getting a great view of Crew 9 inside Dragon Freedom as it returns back to Earth.
Stand by for splashdown.
And splashdown. Crew 9 back on Earth.
It was just fantastic.
I know. I wonder how much solitaire they had been playing for those 9 months. You know, that's an unfair comment, though, because, of course, they weren't playing solitaire. As Sonny said in the news conference, they spent their time up there doing more scientific experiments.
We're doing a bunch of stuff with physiology, understanding what happens to humans as they're in space for a longer period of time. That brings in lots of science experiments in chemistry, in biology.
And there is recent science that talks about how much they aged in space. So both Sonny and Butch aged biologically nearly 40 years.
Oh my God.
In the time they were outside of our atmosphere.
So they got to be a science experiment, an unplanned science experiment. Okay, so there was no design of experiment, but let's make use of the fact that they're spending extra time in space. But look, if aging isn't happening fast enough around us on the planet already, believe
the
me.
mere mortals.
Yeah. Why do we want to know how to age people faster? Surely we should be working the other way around.
I'm glad you asked. And in this episode of Discovery Matters, we are looking into the groundbreaking research that is defining our understanding of health, both in space and here at home.
Okay, so we thought about how the impact of all that time might affect their mental states, but this episode of Discovery Matters is all about how space affects the body.
My name is Colleen Dean, and I'm a lecturer in muscle cell biology at the University of Southampton in England.
Colleen is looking at what happens to muscle mass the longer humans are in space. And she came to this topic in a rather roundabout way.
It all started when I was doing my master's. I was lucky enough to get into a cell culture environment where I was able to get my hands on some muscle cells, and I was really astounded to see how plastic they were when we were giving them different treatments. So we were looking at things like testosterone, which is a steroid hormone, and we know now that that can really have a huge impact on muscles. I went on to do my PhD, very much muscle focused, but it was with my PI at the time, now Professor Tim Etheridge at the University of Exeter, who was in the spaceflight field, and a project and an opportunity then came up, which is now known as the molecular muscle experiment. And it was very much around looking at why do we lose muscle in space and how do we lose muscle in space.
Imagine we've created a world where we can fast forward decades of aging in just a matter of months. What a lovely dystopian place we've created. It's not science fiction, it's the reality that astronauts face today.
The extreme conditions of microgravity cause muscle loss at an alarming rate, and that provides researchers with a unique opportunity to study how and why our muscles deteriorate over time. Colleen was able to work towards understanding muscle decline not just for astronauts, but for all of us here on Earth.
I don't feel so bad about my press-ups now and not doing them, but I think I should get back into it, right?
We know that spaceflight is this hugely negative environment for our health, at least. And we can see that muscle mass and strength can decline by about 20 to 40% in a 6-month stay. If you spend 1 year in space, you lose the same amount of muscle mass in that time that you would do on Earth over 40 years worth of aging. What it gives us is this accelerated aging environment for us to study muscle loss that we hope we can then translate back down to Earth and try to help us maintain healthier muscle mass for longer with aging. Because when we think about aging on Earth, we know that our skeletal muscle declines after the age of 40, and that can be at anywhere roughly around 8% per decade. And we know that although it declines and how important muscle is to our metabolic health and our functional health, we still don't have fully effective non-pharmacological treatments to stop that. So what we do is we try and research muscle health in space to try and improve muscle aging on Earth.
That is where the microgravity muscle experiments, MME and MME2, come in. These studies aimed to uncover the biological mechanisms behind muscle loss in space and explore potential countermeasures to prevent it. So by studying these factors in space, researchers are hoping to develop strategies to preserve our muscle function in orbit and on Earth.
The aim was to try and validate or at least investigate potential countermeasures to this spaceflight-induced muscle decline. We had two working hypotheses. One was around insulin signaling within the muscle. We know that on Earth, insulin signaling is very important for helping us to regulate our glucose uptake, so taking the sugar out of the blood and keeping us metabolically healthy. And it's also very important and also associated with protein turnover, so keeping our muscles healthy and contributing to that muscle mass. But what some previous work had shown in spaceflight is that actually that signaling cascade is reduced. So we wanted to look at that and validate that and see in space what happens to insulin signaling. The second working hypothesis that we had was around integrin attachment complexes. So these are protein complexes essentially that allow forced transduction into the muscle. And we know that this is also associated with, again, things like protein turnover. So trying to keep that muscle mass quite healthy. And similarly, others had shown that actually there may be changes to this pathway in spaceflight. We were also really interested then around the mitochondria. It's responsible for generating energy that we use, and we use that energy to build muscle. We know that in spaceflight, the mitochondrial health declines. So we were then looking at countermeasures to target that mitochondria, again, to see if we can mitigate the loss of mitochondria in spaceflight.
The focus of the molecular muscle experiments might not be what you expect. They were carried out in C. elegans.
Oh my goodness, the mighty nematodes.
Yes.
Okay, but why nematodes? I mean, they don't have muscles like we do, or do they?
They, first of all, were the first animal to have their genome sequenced.
Yes.
And there are many good reasons why C. elegans are the perfect models for these experiments.
They're quite easy to manipulate, but there's also other certain advantages. They have a lifespan of roughly 2 to 3 weeks, so it's very easy to do lifespan experiments in a C. elegans. And also they're translucent, so if you're particularly interested in a mechanism, you can follow it visually because you can see straight through them. Although they certainly don't look anything like us as human beings, the muscle ultrastructure is actually very, very similar. And the genes that make up the muscles of a C. elegans have about 80% similarity to the genes that human muscles have as well. When we then think about spaceflight and actually sending an experiment to space, things that you have to consider are the weight of the payload. It costs a lot of money to send anything up to space, no matter how small. Any project that goes up to space, the astronauts are juggling lots all at once. So it's very competitive to try and get that. So they wanted to work with a model where it was cheap and easy to use. It doesn't take much astronaut time.
There is power in numbers. If you wanted to do rodent research, you might be limited to 4 rats because of the cost and space available in spaceflight. But you can send up to about 36,000 nematodes per experiment.
And you don't have to give them little, like, rat spacesuits or anything like that. And, okay, get it. So, I'd like to revisit the translational learnings that are going to be applied to health on Earth. Because how is this benefiting me as I approach my decades of decrepitude? What are we learning from space for health on this planet?
We've seen that in one of the spaceflight experiments recently that collaborators have done, they actually sent up to space a C. elegans that's very similar to a muscular dystrophy that we have on Earth, known as Duchenne's muscular dystrophy. So they sent up this worm, it's called a DIS1 worm, but it looks like the dystrophy worm significantly loses strength in this spaceflight environment. And actually a lot of the reasons or the genes that were associated with this decline were related to the neuromuscular aspects. So that might give us a promising avenue for looking at dystrophy research on Earth. So we might be able to find, for example, neuromuscular nutrients or drugs and trial them in the dystrophy context. An additional thing would be linking the Duchenne's with the mitochondria, because I think that where we found that in spaceflight the dystrophy worm loses a lot of strength and we see some of the genes that we think are associated with this. We've also on Earth done some investigations using a drug that actually targets the mitochondria, and we see that it can improve that in the dystrophy worm. And so I think that a logical next step is obviously then probably combining that dystrophy scenario with the mitochondrial targeting drug and popping that into the spaceflight context to see if that has any utility and efficacy.
So, considering the UK doesn't actually have like a rocket-based space program, they often have to piggyback off countries that do. So, being part of the first UK-led experiment in the International Space Station must have been quite, an achievement.
This was very much pioneered by the PIs at the time, Professor Nathaniel Shevchuk and also Professor Tim Etheridge. So the PIs invited on collaborators from Japan, for example, and also Greece. So we weren't just looking at our own hypotheses around muscle and the things that change in spaceflight, but actually some of our collaborators were looking at the role of dopamine, for example, and how that can impact adaptations to spaceflight. So the actual project in and of itself was much bigger than just kind of the UK-led muscle aspect. But yes, it was very much pioneered by the UK. And I think that was really quite special, and seeing it live when you're there and you see your little worms go up on was probably the best part.
All of us care about aging and maintaining health span as long as we possibly can. So as we learn more and more about spaceflight and how that influences aging, the more that we'll be able to actually think about ways to manage it better or things we can do to intervene.
Now meet Susan Bailey, one of the lead researchers on the NASA twin study.
I'm professor of radiation and cancer biology at Colorado State University in Fort Collins, Colorado.
The NASA twin study was groundbreaking research that examined the effects of space travel on the human body by studying identical twin astronauts, Scott Kelly and Mark Kelly.
There are 4.5 million twins living in the U.S., but there's actually only one pair of identical twin astronauts, Scott and Mark Kelly. The Kelly brothers have become crucial into understanding what the long-term effects of living in zero gravity are for the human body.
Now, it was Scott who spent 340 days aboard the International Space Station, and that was in the year 2015 to 2016. Mark stayed on Earth. He was the control subject. They are identical twins, similar in nature and nurture. They had both been Navy test pilots, then astronauts. So Mark was actually the perfect control. Doesn't get better than that.
That is very good. So just pause here for a second. As the mother of identical twins yourself,
yes, I am.
Dodie,
that's right.
Would you think that sending them into space, one of them, and I mean, is this a thing you would encourage? Is this behavior that you're really looking for in your twins?
Well, Mama thinks that, they're actually, I'm so glad you asked, because now I'm going to brag. I'm going to brag.
Okay.
They're both training to be commercial pilots. That's exciting.
Okay.
I think the 30,000 cruising altitude is high enough. If you ask me.
They might just age just a little bit faster.
Yeah, they might.
So what actually led Susan to getting involved in this study in the first place?
I am classically trained as a radiation cytogeneticist, so interested in radiation biology and what radiation does, both to cause cancer, how it damages DNA and causes cancer with low doses, but also on the flip side of that, how high doses can be used to kill cancer cells and be used for radiotherapy. I was kind of tapped into the NASA framework, and the opportunity for the twin study came along. And so I applied. I had already had a concurrent study that was looking at telomeres in 10 unrelated astronauts. I had proposed looking at telomere length dynamics and chromosome aberrations or DNA damage in astronauts. The twin study opportunity came along just exactly the right time for me, that I was able then to become part of that and do a much more in-depth type of study with the twins, Scott Kelly and Mark Kelly.
Scott Kelly will be conducting numerous experiments with a rotating crew of astronauts, but his primary mission during his arduous year-long assignment is to study the challenges long-term exposure to the extremes of space have on humans.
It really was a great experience, and it set us up too for doing our current study. So we're also part of what they're calling CIPHER, which is really another NASA study very much modeled on the twin study, in that it's 14 different investigations of all kinds of different things on astronauts on various duration missions. We so far have a number of 6-month crew members, but we're hoping to get another year-long mission and maybe a crew member or two from that.
Aha, the return of the telomere. And that is not a monster from Doctor Who.
They are the ends of our chromosomes. They cap the end of a DNA molecule, the end of the chromosome, much like plastic aglets at the end of your shoestring.
Aglets is a killer word in Scrabble. And telomeres, yes, they get a little flappy and ragged as you age. They keep the end of the DNA from fraying and degrading so that we don't lose any important coding sequences.
Exactly.
As cells divide and as we age, telomeres shorten, and that's just because they can't replicate to the very end of a linear DNA molecule. And so that's why they are associated with aging. The more quickly that we lose telomere sequence, the more quickly we might be aging or developing dementias, are all age-related pathologies that we think about that have been associated with short telomeres. And if you think about space flight, if crew members develop something like dementia or cardiovascular disease, or even some forms of cancer like leukemias that can form fairly quickly in the period of a year or more, those would really be a threat to the success of the mission, but it would also be long-term health effects for the astronauts. Radiation exposure is another one that has been shown to, at least some scenarios, shorten telomeres. That really led us to hypothesize that space flight would certainly act to hasten telomere shortening and perhaps also the occurrence of some of these age-related pathologies that could be very detrimental to astronauts, whether during a mission or after, once they return.
What Susan and her team actually observed was a little different to the hypothesis.
What we found, and not only in the twins, but also in the other astronauts, is that average telomere length during spaceflight was actually longer. It was not shorter, as we've imagined, but it was actually longer during spaceflight. And it stayed elevated for the entire year that Scott Kelly was in space. Then upon return, telomere length shortened very, very dramatically. And that was really and truly something we saw in all of the astronauts. for whom we had the entire set of samples for and telomere length measures. And then over the course of recovery with Scott Kelly, there was some recovery of average telomere length, but the interesting thing was at the end, they did have more short telomeres than when they started.
NASA tonight suggesting that year in space activated so-called space genes. And tonight, Scott Kelly joking, I no longer have to call Mark my identical twin brother anymore.
In the long term, when we think about the astronauts when they return, I do think that that's probably where the long-term health effects are going to be seen.
But it sounds like being in space is a bit of the fountain of youth while you're out there. So you feel kind of young. And I mean, this is, a little bit like the portrait of Dorian Gray. It's like you send yourself up to space, you feel all young there in the attic, and then you come down and you're old.
That's right. I thought the same thing. And so I asked about that. And it seems the elongation of telomeres during spaceflight could be linked to chronic low-dose radiation exposure in space. Now, that's different from Earth's radiation environment. Radiation continuously destroys radiosensitive white blood cells, prompting the body to replenish them with stem-like cells that naturally have longer telomeres. Now, the long-term concern is that excessive stem cell depletion could lead to premature aging-related consequences. So you're going back and forth there between perhaps not biologically age a bit.
What's the opposite of age
young.
a bit? Age a bit, youth a bit.
Right.
Yeah. Okay. I'm youthing today. So what about other differences between Earth and space, like gravity? And
gravity. I've heard of that.
Radiation and lack of art museums. Is there a sense that actually Earth gravity has more of an impact on telomeres dynamics than microgravity?
We've wondered about microgravity because that certainly is the other obvious thing that astronauts are experiencing during space flights that we don't experience here on Earth. And at least as far as we can simulate microgravity on the Earth, we have not seen changes in telomere length with microgravity alone. The great thing about telomeres and being able to look at them during spaceflight is that it incorporates all of the stresses, the microgravity, all of the radiation exposure, all the psychological issues. The bad thing is that it's very difficult to tease out any one of those particular stressors, particularly on Earth, to try to simulate any single thing that might be contributing to those changes is very difficult. And a lot of that is because we can't do some of these more space-relevant radiation exposures for months at a time. You can't really replicate the doses and the dose rates that you see in space.
So only about 600 people have ever traveled to space. The vast majority of astronauts over the last 60 years have been, guess what, middle-aged white men on short-duration missions of fewer than 20 days. So that's a relatively homogeneous cohort. Does this new study have more diversity to it?
The idea behind our involvement in CIPHER was exactly that. We could start to look at a larger, more diverse population of astronauts because certainly we have observed individual differences in response. There were individuals who either did not experience the same thing at all or it was much more extreme. At the forefront of why we're doing the current study, hoping again to get more women, at the very least more women, which we do have in the new study, as well as different ethnicities and races. Their plans are to get to the moon and then on to Mars.
Susan said earlier that the foundations from the first twin study meant that the team could better understand which questions to ask in the second study. So what questions are they? And again, how does this relate back to little old me and our science back on Earth?
The insight that we have so far as to what might be influencing telomere length. Oxidative stress is one. We also are specifically looking at different populations of T cells, of our white blood cells, to see where telomere length changes are occurring. And pretty much similar to what we imagined and have seen so far, we are seeing telomere elongations in particular subsets of white blood cells. We're running those particular panels so that we can actually evaluate how they change during spaceflight. Antioxidants would be fairly effective at trying to slow down that process. Senolytic drugs have also been proposed to target senescent cells. So senescence is very much related to critical shortening of telomeres and cells stopping to divide and promoting aging and age-related diseases. There also are many studies for radiation mitigators that might protect against radiation damage. As we go forward, I think we can even think of space as a kind of a model of aging, because if it's accelerating aging, we can perhaps get some of these answers more quickly than we would otherwise.
Space travel, no longer just for astronauts. Commercial entities are now launching civilians into space, of course.
And nematodes. I mean, just think
Right.
what's going on in the nematode community. They
Get
must be
your
like,
tickets now.
Exactly. Have you been to space yet? Well, 36,000 of our buddies did.
Buy 35,000, get 1,000 free.
So this is a really exciting shift in space exploration. And what's really intriguing is how these civilians with no formal training react to the effects of spaceflight, because of course they're not muscle-bound test pilots.
4 civilians set to launch into orbit for a 3-day voyage circling the Earth. This is the first time anybody has ever put civilians into orbit.
One of the first ones that had an all-civilian crew. They had the youngest astronaut, the young lady that was a childhood cancer survivor. It was just a remarkable thing. So that is a very interesting question, too. How do these untrained civilians react to spaceflight? And in fact, we were able to look at telomere length for that inspiration for crew. And it was only a 3-day mission. It was a little bit higher in elevation than the space station, so it was a little higher doses and dose rates than we see on the ISS. But we saw telomere elongation in 3 of the 4 individuals. So even after just 3 days. So we've seen everything from 3 days all the way up to a year. So the finding is becoming very consistent. Now our challenge is to figure out why.
I'm Rihanna Bokhari. I'm the Scientific Research Director at the Translational Research Institute for Space Health. I study space health, and so the Translational Research Institute for Space Health is a partnership with the NASA Human Research Program. We're tasked by NASA to do things a little bit differently, but still attack the NASA human research risks, but do them in a slightly disruptive or take a different angle to try to solve the problem.
So what does that mean, to be disruptive in an ecosystem when you're leaving the Earth's atmosphere?
The industry of space in general is disruptive just by its nature, but NASA has a human research roadmap. That's this integrated plan to reduce the major risks of human spaceflight. And so NASA is really taking their stepwise approaches to try to find different ways to reduce the risks and attack those risk reductions in ways that are just outside the box thinking. So we're supposed to do high risk, high reward work and do things that might fail, but at least we learn something. We funded a group of projects on metabolic manipulation, trying to reduce the metabolic rate that people would experience so that we can reduce their resource utilization on the way to Mars or any future location. We measured all the different effects that happen to the physiology, and we found that they were able to reduce their oxygen consumption rate and all the things that you might worry about in terms of resource utilization.
When we talk about long-duration space flights, are we looking at something like hibernation pods or like sleep boxes?
It's really tempting to think that it's like the movies. It's not quite like that.
I want it to be like the movies.
I know, but they're definitely exploring innovative concepts along those lines.
Obviously, folks have wanted to explore. Mammals can hibernate. It's potentially in our genetic memory to be able to do that. Trying to understand how the body could do that to reduce our needs in terms of our supply needs would be hugely beneficial. Maybe they're not necessarily hibernating like a bear would, but maybe they're just in a resting state and using less resources during that time period. Maybe even sleeping more, just to make sure that you don't get as bored. You can imagine a travel that's 6 months long where you're stuck in a tin can with 4 to 6 people might be mental health stress, and you might want to just sleep through most of it. These are strategies that we've considered, and that's a little outside the box for the NASA world, but we found some interesting data in terms of how that metabolic manipulation might actually benefit us from an operational NASA perspective. Another area that we've recently explored is how to make endogenous repair better, our capability of fixing the damage. And improving that endogenous repair might be a great way to just overall reduce our risks. Now we're getting towards the latter part of our cooperative agreement with NASA. We have started to focus on autonomous healthcare.
And so what does Rhianna mean by autonomous healthcare?
Right. So if you compare it to the way it's done now, when an astronaut is given healthcare on the International Space Station or in any low Earth orbit mission, the doctors on the ground and all of the healthcare treatment plans and actions require back and forth communication.
That is not a tenable way to handle things for the Mars context, right? Because Mars has a communication delay up to 20 minutes each way. So if you had to call and say, hey, what drug do I take for my headache? And then it takes 40 minutes to get a response. Well, that might be fine for a headache, not ideal, but it might not be great if you have like a broken bone or something much more serious where you need much quicker intervention. And so Trish has been really focused on trying to think about better ways to think about autonomous healthcare. And so innovating in that space just to try to create the network of capability to plug in tools for autonomous healthcare has been the first step of that journey. And we hope to plug in even greater tools in the future.
One of the key takeaways from this conversation was how the advancements in autonomous healthcare for space missions could revolutionize care here on Earth. It's all about using data and technology to improve health management, not just reactively treating illness, but proactively preserving health and preventing problems before they escalate. The stuff that insurance companies love, right? And this could be a game changer, especially for underserved communities in rural, remote areas with limited access to healthcare.
The reason autonomous healthcare is actually helpful for us here on Earth is because we see hospitals closing left and right, and there are many people that are not close to a hospital or in a rural community where access is limited. Because right now, you might have a wearable device like a Garmin or an Apple Watch or something like that, and that's collecting constant data. And how we use that data right now in a healthcare context is usually pretty limited. But there are other types of monitoring devices like continuous glucose monitoring and other things that are in development right now. It's continuous data on the same person over time. And so learning how to use that data to help inform a person's health and health state would help maybe transition us from having a reliance on just treating illness to moving towards a preservation of health, which might actually improve our capability of living longer better.
So I love this, but this personalized healthcare, how does it translate to astronauts who are facing a really unique set of challenges out there in the darkness of space?
When we think about going to Mars and carry only what we need, we actually want to probably think about personalizing the medical care for each individual astronaut. We want to make sure we have the drugs that they will respond to, make sure we have the specific types of things that they might actually need based on their personal risk profile. NASA government astronauts tend to be extremely healthy people, but everyone, as they progress in their life, develops health risks, right? So the whole goal is to make sure you are enabling the capability of anyone to go do this. And as we start to be able to do that better and personalize that approach, maybe that means even more people can go into space safely. I think that personalized medicine approach will also be an area that has a lot of interest for biotech and is of great interest in the NASA space. And hopefully NASA can make some progress there that maybe will help us here on Earth.
With that kind of progress in personalized medicine, we might not just be talking about space travel. This could very well pave the way for extraterrestrial colonies one day far into the future.
I certainly hope to see colonies in the future. I don't know if 100 years is enough time. It's taken us a long time to figure out how to reduce the risks for any one travel piece, but I think we already have lived for decades. So we've made huge leaps and bounds in the years that we have had the space program. And so I expect we will continue to progress in that direction. Don't know if 100 years is enough, but in a couple hundred, I could see for sure we would have colonies in other places. It's human nature to explore and expand.
Connor, are you up for a timeshare on Mars in 100 years or so?
Do you know what? I'm a little bit conflicted. I feel like humanity's acting a little bit like a petulant child that's like messed up one of its bedrooms and it's like, let's go work in another one. And it's like the clean one. Honestly, I would, however, think I'd quite like to grow mushrooms like in that episode we did a year or so ago for my furniture on Mars. I mean, that could be quite fun.
Absolutely. So for that kind of faraway future, there are concerns about the radiation that we were talking about earlier.
Some people may have heard of something called the Carrington event, which is one of our largest recorded solar particle events, and that caused electronics to go out here on Earth. It's enough to cause some damage, but it's not really enough to cause death or anything like that. It's from an acute radiation standpoint, that's probably enough to cause some damage to the blood-forming organs, so like your bone marrow. But if you get to a hospital quickly enough and get treatment, you should not have any issues, maybe a slight increased risk of cancer. And so we worry about those in space. For the other kind of radiation that's in space is called galactic cosmic radiation. It's basically from celestial bodies or supernovas that come from outside of our solar system, but there's about 1% of that spectrum that is from high energy particles. So these are like high energy heavy particles, and when they pass through shielding, they create more radiation on the other side. And that's a real problem. We can't recreate it here on Earth, so we really need to go back outside of low Earth orbit to really understand the biological effects of that kind of radiation. NASA tries to keep the increased risk of cancer to below 3% increased risk of cancer in the lifetime. And right now, for Mars, it'll be likely more than that. That's one of the big risks.
Radiation exposure in space is a completely different animal compared to what we see in using radiation in cancer treatments, for example, here on Earth. Yeah,
totally. And it's also not the same as the occasional radiation we get from solar events here on Earth, like the Carrington event. So while those can cause some disruption, the kind of radiation astronauts face in space is much more intense and persistent.
What an episode. These conversations are just out of this world. Oh, sorry, that's bad. That was like a Dodie level pun. I'm really sorry.
That absolutely was. Congratulations. Welcome to the low-level world of puns. From muscle plasticity to spaceflight-induced aging, Colleen's research showcased how studying space can deepen our understanding of health here on Earth.
And then the fact that spending a year in space actually ages you 40 years in your muscles on Earth is just kind of mind-blowing. And the impact on our understanding of conditions such as muscular dystrophy, I mean, that's just huge. And the idea that mitochondrial targeting therapeutics could benefit not just astronauts on long journeys, but also people on Earth really highlights how space research and exploration really extends the science sense of what's going on down here on Earth.
Absolutely. And Susan's work on telomere dynamics in the NASA twin study gives us valuable insights into accelerated aging.
And learning from Rihanna about the efforts to mitigate risks for long-term space travel was absolutely fascinating. Trish is taking this bold, disruptive approach to exploring high-risk, high-reward research to improve astronaut health and safety.
We learned a lot this episode, and every day is a school day, so now let's learn even more.
So, Dodie, what have you learned this week that's blown your mind?
Well, it's also been about space, and it's been about getting clues about life from billions of years ago.
Okay.
So, NASA made an amazing discovery on the asteroid Bennu. They sent a spacecraft
piloted by little elegans. I cannot get away with this picture of little worms piloting our spacecraft.
I think unmanned. But Bennu is about 200 million miles away from Earth. And the aim was to collect samples from the surface of this asteroid, gathered a handful of dust and rocks, brought it back to Earth, where they put it in a protected environment so that then that dust and those rocks wouldn't be polluted by Earth. Exactly. And they analyzed the samples and found molecules that are essential for life. Amino acids. You recognize those as the building blocks of proteins on the asteroid.
Insane.
Yes. So suggesting that the ingredients for life might be more common in the universe than we thought.
So lending credence to the panspermia theory.
There is life out there.
Wow.
Yeah.
Or it may have originated out there.
Correct. Could help us understand how life on Earth began.
Mm-hmm. And is it intelligent life? Because there's none down here.
Beam me up, Scotty.
Amazing.
What did you learn this week?
Well, I'm very excited because of recent research that has highlighted a potential link between the common and garden variety shingles vaccine and a reduced risk of dementia, including Alzheimer's.
Wow. So shingles related to chickenpox.
Exactly. So if as you get older and your immune system forgets a little bit its reaction to the chickenpox virus, you can experience shingles, which is really, really unpleasant. A recent study published in Nature found that the live attenuated shingles vaccine reduced dementia diagnosis by an extraordinary 20% over 7 years in a Welsh population. And another study showed that the recombinant shingles vaccine, so it may offer even greater protection against dementia. And then there's research that also suggests that herpes viruses, such as the very common herpes simplex 1 virus that, we all got kissing behind the bike sheds at school, they may contribute to Alzheimer's disease by activating harmful genetic elements in our brains. So while the shingles vaccine role in Alzheimer's prevention is really promising, we need to do a lot more research to really understand the mechanism and its effects. So it's very exciting. We'll link to the Nature study in the show notes.
Coming back again to that classic scientific conundrum, the more you know, the more you realize you don't know.
Yeah, and then, who knew? We've been looking at the amyloid hypothesis and tau tangles as the cause or, one of the effects of dementia, but very few scientists had pursued the idea that it was actually potentially a virally triggered condition. Amazing.
Our producer is Beth Armit-Brewster, editing, mixing, supervision by Tom Henley. My name, Dodie Axelson.
And I'm Conor McKechnie.
You are.
Make sure you rate us on Spotify.
Please.
Or whichever platform you use. We'll see you when we get back from Mars with a whole bucket of C. elegans and another episode of Discovery Matters.